Hello, I'm Paul Beckwith. So, initial reports were saying that the coronavirus, the R0, which is, if one person has the virus, how many people do they spread it to? The R0 was R at time 0, the initial spread was 2.4, but a paper that I discussed earlier says that that number is more like 4.7, which is huge, huge transmission. So I'm going to talk about, what is the science behind why the novel coronavirus is so contagious. So at the end of the last video, I started talking about this paper. So just Google the title. It's in the Medium. What makes it so contagious, and it's the features of, prion-like features, which make it 20 times more likely to stick to protein receptors found in human cells. That's the ACE2. And this contributes to the human-to-human spread of the COVID-19. So basically, how does it get into our cells? Okay, first of all, the name coronavirus, it comes from the Latin word for crown or wreath, which is the visible structure of the viral particles. Viral particles, the term virologists use is virions, V-I-R-I-O-N-S. But this is a cross-sectional, so this is a 2D image. And what you can see is that you can see these spikes here. Okay, this is an electron micrograph of bronchitis coronavirus-like particles. So the diameter here is maybe, somewhere like 50 to 200 nanometers is typical for viruses. Very, very tiny critters. A nanometer is a billionth of a meter. This is a 3D image of what this is an artist's rendition of a coronavirus, so we all know about this. Most people are aware of this shape now, a sphere with these proteins sticking up, these spikes. Okay, these spiky protrusions are called spike proteins, and they're very important in replicating coronaviruses. Okay, so the virus is not alive. It's a self-assembled collection. It's got a lipid outer surface. It's got proteins in the lipids. It's got RNA, ribonucleic acid inside, which is one of the branches, one of the spirals in a DNA strand, and it's got the spike proteins which allow it to attach to the cells in the body. Now, viruses, they don't make copies of themselves like most living things, and they're not living, right? It's this collection of things I just discussed. But it takes over the cellular machinery of other life forms and it sort of hijacks the infrastructure to replicate itself. So it's got a very small genome. So the number of gene pairs is very, very small. Okay, it doesn't encode it doesn't need to be complex. It doesn't do any of the stuff that living animals do when they replicate, DNA replication machinery. There's very simple instructions, very few simple proteins. So in the human genome, right, there's several million distinct proteins, but there's only 28 proteins in the genome of the coronavirus that causes, the disease. So one of these proteins, one of these spike proteins, binds to receptors on a cell of its host. In human, the receptor is called ACE2. Angiotensin converting enzyme 2. It's just a receptor. It's like a docking station. There are medications that treat high blood pressure and other disorders. They're called ACE inhibitors. Okay, so maybe they, some variant of them can be tried, to make different vaccines. Okay, but the ACE2 is just a docking station. Okay, and then, so when the virion binds to those receptors, then there's a process called endocytosis where it enters the cell. Once inside the cell, it releases the RNA material, the genetic material, and the cell starts making copies of it, these copies leave the cell and repeat the cycle. So this is the coronavirus. Right, this is a cross section. So you've got a sphere here. You've got the single strand of RNA inside, and you've got these spike proteins here. This is the lipid and proteins making up the fatty outer layer, which annihilates. Okay, so it goes to the cell of the body. There's an ACE2 receptor docking station. The spike makes contact here. Then this thing kind of splits open, and the RNA can go right into the cell where it does its hijacking, and it does its nasty work within the cell and replicates itself. Okay, so the ACE2 is necessary and sufficient for the infection to spread. Okay, the most common comorbidities, so people have diabetes, hypertension, and cardiac diseases. These disorders that are often treated with ACE inhibitors, then you're much greater risk of being seriously ill from the coronavirus. But the virus can be blocked from entering cells by preventing its binding to ACE2. Okay, so it's the spread, the transmission of the coronavirus is enormous, and it's driven by its ability to bind to ACE2. Okay, so if we could stop that happening, then that's what that's what the vaccine or natural antibodies that build up will take a role in doing. But the problem is the story is incredibly complex. I hope I'm not botching it up too much. If there's higher levels of these ACE2 receptors, then it's easier for the virus to enter the cell. Okay? But it also makes us more likely to survive and thrive as a result of the infection. If there's lower levels of ACE2, then it's harder for the virus to enter the cell, but if it does, then it's a lot more deadly. Okay, so they say it's kind of freakishly intelligent, or it seems like it is, the coronavirus. Okay, it's like they're intentionally targeting a protein that plays a role in fighting them off. Okay, so by using the ACE2 as an entry, there's a balancing act. If we downregulate the levels of ACE2 to lessen the risk of the transmission of the viral infection, it leaves us susceptible to the damage caused by an infection. If we upregulate levels of ACE2, which makes the infection more likely, it helps mitigate the resulting damage. So it's a catch-2 between deadliness and contagiousness. Okay, so, and I've said before, this virus is in the sweet spot between these, because if a virus is too deadly, it kills the host before the host can spread it. And also the symptoms appear much earlier. But for this guy, many people are asymptomatic, and the people that do get sick can be asymptomatic for up to 2 weeks. So how do you stop something like this? This is SARS, small number of cases, high mortality. MERS, even smaller number of cases, much higher mortality. Swine flu, huge number of cases, very low mortality. So usually there's an inverse relationship between these. COVID is in the sweet spot. These numbers are continually updating. Okay, so the viruses ensure their survival by maintaining an evolutionary arms race with their multicellular hosts. They force our bodies to choose between the risk of death or the risk of infection. Okay, so if there's not many ACE2 receptors, the risk of infection is low, but the risk of death is much higher if you do get infected. If there's a lot of ACE2 receptors, and the risk of infection is much higher, but the risk, the death rates are much lower. So, the viruses can respond by rapidly evolving in the other direction to what our bodies are doing. Okay, so this is showing you, this is comparing cases and deaths again to SARS and MERS. It's a rapidly evolving situation. We're learning lots about it. But so here they looked at amino acid sequences of the proteins of the coronavirus spike to see how it differed from other coronaviruses. Okay, and the key thing is there's an intrinsically disordered region, and I'll explain what that is. But first of all, let's talk a little bit about proteins. So the primary structure, when proteins are first translated from RNA, they come out as a simple linear chain of amino acids. So the amino acids are the building blocks of proteins, and you get this chain here. This is the primary protein structure. Then what happens is there can be cross links between different. So this is one link here, another link here. There can be cross links between them, or there can be a helix. There's hydrogen bonding of the peptide backbone, causing the amino acids to fold into a repeating pattern. And then there's these structures can then interact with each other and create a tertiary structure known as a folded polypeptide. And then the protein, a bunch of these guys can combine. There's one of them, there's another one. They can combine and interact with each other in these large complexes called proteins. So they need to be folded into the right shape to do the proper thing. So hemoglobin, for example, proteins carry oxygen in our red blood cells. Without hemoglobin, we would suffocate. So there's 4 polypeptides folded. One change in the amino acid sequence can change the structure, and this means if it's misfolded, folded the wrong way, then you get sickle cell anemia, a powerful and a painful and incurable illness. So protein folding is key for the function of the protein. So this is normal hemoglobin. You've got 4 of these clusters here, the quaternary structure. This makes up a red blood cell. The red blood cells move through and they carry oxygen, after they're oxygenated in the lungs. The sickle cell hemoglobin these guys here are connected together, and it forms like a sickle shape, red blood cell, and then these things can form clots and blockages, and they don't carry oxygen properly. Okay, so it all is to do with the folding of the protein. Now, this is what happens. This is a structured protein. Think of it as spaghetti that's not cooked, so it's hard. Throw it against a window, it doesn't stick, it bounces off. This is an intrinsically disordered protein. This is like cooked spaghetti, soft. Throw it against a window, it'll stick. This is a structured protein with a disordered region on it. So this is what the coronavirus is like on the spike. And what this means is that this can stick easily to cells in the human body. Okay? So, it's like a dumb game that you can play, throwing noodles at a wall. A piece of uncooked spaghetti is rigid and will bounce right off the wall, but a well-cooked noodle is flexible and will stick firmly. So the coronavirus spikes, the protein spikes, are of this nature. They have a wet noodly structure which allows it to stick to cells. So it can bind tighter. The tighter it can bind to our cells, the more easily they can affect us, the fewer viral particles that are needed to cause an infection. The person, so in the game of spaghetti throw, if you ever played that, I did, the person who sticks the most noodles to the wall or window wins. So here she goes. She's throwing, spaghetti. If it sticks, if it's wet, it'll stick. Of course, uncooked, rigid, it'll just bounce off. Okay, so so it combined. So what are prions? Well, prions are, you can think of them, in the brain. They cause these disorders. They can be. So here's, this is a normal protein. It can attach to, this is the damaged, the misfolded prion protein is red, and the normal proteins are here. So the normal ones get captured, if you like, and lead to abnormal ones. So it messes up with the folding, and it's the reason why the virus is so transmissible and so small number of particles cause infection.